Welcome to week 2. This week we're going to talk about blood cells. The corresponding unit is unit 20 in your lab manual and also refer to chapter 18 in your lecture text. Let's talk about the functions of the circulatory system. So in this lecture, this lab lecture, we're going to focus in on blood, but it's important to know the function of the circulatory system as a whole. So the circulatory system consists of the heart, the blood vessels, and the blood. We're going to focus on the blood. The cardiovascular system refers only to the heart and blood vessels. So that's what we're doing next week. We're doing the cardiovascular system next week. We're not going to get into a lot of the blood vessels. We will get into some of the blood vessels, but a lot of that's going to be actually after this next test. So hematology is the study of blood. The functions of the circulatory system. Big function here is transporting that oxygen and CO2, nutrients, waste, and hormones and stem cells. Our tissues need oxygen and we need to get rid of that CO2. So another one is protection, inflammation, limit the spread of infection. We're going to talk about white blood cells today, and they're gonna destroy microorganisms and cancer cells, neutralizing toxins and initiating clotting. So we're going to talk a little bit about platelets, which have a big function in blood clotting. That is their main function. Also regulation of fluid balance. So we're going to stabilizing the pH of the extracellular fluid and temperature control as well. With the circulatory system, we can constrict blood vessels or dilate blood vessels to allow your blood to actually go to the surface of the skin so that we can closer to the surface of the skin so we can actually get rid of some heat or we can constrict it if we want to conserve heat. So now let's talk about the components of blood. So adults have 4 to 6 liters of blood. It is a connective tissue, which consisting of the cells and the extracellular matrix. So there's two components. There's the plasma and the formed elements. So the plasma is that matrix, that extracellular matrix, and the formed elements, those are the cells. So the plasma, that's the clear light yellow fluid. We're going to take a look at that when we talk about hematocrit. And the formed elements are actually the cells, the blood cells and the cell fragments, including those red blood cells, the white blood cells, and the platelets. So there are 7 kinds of formed elements. So here you can take a look over here. This is kind of an exaggerated image. So if you're looking at a slide of blood, you wouldn't see this many white blood cells. So for this illustration, they just wanted to get as many of all the different white blood cells in here. But if you're looking at a slide, the most numerous formed element is actually the erythrocytes, so the red blood cells. And then you also have your platelets, so right here. So they're very numerous. And then the least numerous are actually the leukocytes, these white blood cells. So leukocytes, it's just another name for white blood cells. So number 1, erythrocytes. Number 2, we have platelets. And the main function that you need to know for platelets, just know that their main function is blood clotting. So the leukocytes, we have 5 leukocytes. They're divided into 2 categories, and we're going to talk a little bit about how we remember that. But you have your granulocytes. These have the granules, and we'll go over what granules are in a little bit. But neutrophils, eosinophils, and basophils, if you watch my other video, these are filled with, they end in PHIL. They're filled with granules, so then you can keep it straight that the neutrophils, eosinophils, and basophils are granulocytes. And the agranulocytes are without granules. So those are the lymphocytes and monocytes. And I have some mnemonics for you to help you remember that. So now let's talk about hematocrit. So we actually do this in lab where we prick your finger and then we have a capillary tube. We actually get the blood to go into the capillary tube and then we put it in a centrifuge and we spin it around and then it comes out and it looks just like this. This allows us to separate the different components of the blood. So you'll be able to see the erythrocytes are on the bottom here because they're the heaviest. So we would put them in the middle. So everything, all the heaviest elements go down here. So you have your erythrocytes right there. And it's about 37 to 52% of the total volume. And in lab, we actually have you measure it. So you'll take a ruler and you'll measure from here to here. You'll measure your hematocrit, which is your red blood cells, and divided by the whole volume. So you take this divided by the whole volume of everything, so including your plasma and your buffy coat, and that's your hematocrit. So the normal is about 37 to 52% of the total volume. White blood cells and platelets, so those are the white blood cells and platelets are in this buffy coat here, so it's really small, and it's hard to measure. Depending on your the size of your blood sample, you should be able to see it, whether it's really noticeable or not. Depends on the size of your blood sample. And then you have your plasma. So your plasma is up here. This is about 55% of whole blood here, 47 to 63%. It's a complex mixture of water, proteins, nutrients, electrolytes. So you can see how different things could affect this. So if you're like super dehydrated, your plasma levels would be decreased. So you're going to get a different number for your hematocrit because you'd be taking this divided by this. But if this is only here, you're going to get a higher number, a higher percentage. So if you're super dehydrated, your hematocrit is going to be elevated. On the other hand, if your red blood cell production, your red blood cell count is down, then you're going to get a smaller number because you're going to have less red blood cells. Also, we have the leukocytes here as well. And I think you really need to know how to calculate this. So you definitely need to know how to calculate the hematocrit for whether that be lab quizzes or tests. There will definitely be some questions, at least on the lab exam. So make sure you know how to calculate the hematocrit. So this is an actual test tube that we completed in class. So you're going to measure the entire blood sample. So from here to here. So here's the plasma, there's the buffy coat, and here's your erythrocytes here. You're going to measure that entire sample. Here's a wax plug, so you're not going to measure that down here. And then you're going to measure the cells packed towards the clay bottom. So here's the clay bottom here. You're going to measure from here, the red here to here. So measure that red. And then you're going to divide the cell measurement by the total volume. So you're going to go this number divided by the whole sample. And then you're going to get that. It's going to be a 0.45, for example, and then you're going to times that 0.45 by 100 to get your percentage. So then you get 45%. You're going to record, well, you're not going to, but we did in the face-to-face version record your hematocrit on a class data sheet. So that's how you calculate the hematocrit, the PCV. These are the packed cells at the very bottom here. So you take the red divided by the whole sample size, and then you have to times it by 100 to get a percentage. So this is what a blood slide looks like. So underneath the microscope, it looks like this. The most numerous formed elements on here are the erythrocytes. You can see there's tons of them. We also have some white blood cells and we have some platelets here too, but the most numerous are the erythrocytes, those red blood cells. So the two main principal functions, so number one, we need to carry oxygen to get it to the tissues. Number two, we need to take the CO2, that waste product from the tissues, and bring it to the lungs so we can get it out of the body. So that's the main function right there. So that's why it's important it's very important that's why we want to even know what the hematocrit is, because if there's not enough red blood cells, this can cause death in minutes due to lack of oxygen to the tissues, because the main function is actually transporting that oxygen and getting that CO2 out of the tissues. So that makes sense. So that's why it's important. That's why we want to know what the hematocrit is. So I don't care if you know, you don't need to know the exact diameter, but just know that the erythrocyte looks like this. and they lack mitochondria, so they're not going to be stealing. They're not going to be stealing the oxygen. If they're carrying oxygen, we don't want them to take the oxygen for themselves. So they're going to actually do anaerobic fermentation to produce ATP. They're not going to use oxygen to produce that ATP. Otherwise, your red blood cells would be stealing that oxygen, which you need to bring it to your tissues. And they also lack a nucleus and DNA, which makes it very difficult for them to actually, they can't repair themselves. So they have a very shortened lifespan of around 120 days. So your red blood cell count and your hemoglobin concentration indicate the amount of O2 blood can carry. So what is hemoglobin? It's a protein in erythrocytes which binds and transports oxygen. So here are the tests that we did. We did the hematocrit. So that is one of the main tests we do in lab is hematocrit, which is your same thing as PCV, your packed cell volume. So the percentage of whole blood volume composed of red blood cells. And important, I do need you to know that these numbers. So men are, it's 42 to 52%. That's a normal hematocrit for men. And for women, it's 37 to 48%. So also another thing we do in lab, we do the hemoglobin concentration of whole blood. We do a hemoglobin test. Once we've gotten our hematocrit, we got enough blood into our capillary tube. If we have some left over, we put it on a piece of paper called a talus paper, and you put it on the piece of paper, and then you have a chart where you can compare the color of the blood to this chart, and you can see if it's not where it's supposed to be, the correct color, then it could be indicative of an anemia, such as, well, specifically iron deficiency anemia. So I don't need you to memorize this, but I really want you to know this and just be able to understand how we can test hemoglobin concentration. Also red blood cell count, so we don't do that. That's in a lab. And so here are the numbers for that. You're going to notice, well, you notice that men typically have higher counts. They have higher hematocrits typically. Well, actually, obviously they do. And they have higher red blood cell counts and a higher range for the hemoglobin as well. And we're going to talk about that, why that is. So we just saw how the values are lower for women. So why is that? So the few reasons here. So androgens stimulate red blood cell production. So androgens are male sex hormones, and males have more of these hormones. So therefore they're gonna have more stimulation of red blood cell production. Therefore the hematocrit's gonna be higher. Women have periodic menstrual losses, so they are bleeding. They will be losing blood. Hematocrit is hematocrit is inversely proportional to percentage of body fat, and typically women have a higher percentage of body fat. So let's talk about an excess of red blood cells, which is polycythemia. So if you have increased red blood cells would be polycythemia. If you have decreased red blood cells, that would be anemia. So here are some causes. I don't need you to know, for lab, I don't need you to know primary or secondary. I just want you to know that some reasons why you can have excess red blood cells. So if you're dehydrated, your hematocrit is going to be super high, because if you're dehydrated, we're going to decrease that plasma volume, which is going to, you're going to divide the red blood cells volume divided by a smaller volume, so you're going to get a higher percentage. High altitude can cause this. Physical conditioning. So if you are working out, that's why athletes will train at high altitude to increase your carrying capacity for oxygen. There's some dangers though, and that's why Lance Armstrong, I think he was caught with blood doping, I believe, which he was injecting blood, his own blood. So he would take blood out of him, and then he would save it, put it in a refrigerator for a while, and then he would put it back into him the day before the race, or I don't know how much time before the race, but then he would have more red blood cells in his body, so then he could carry more oxygen. And so if you can carry more oxygen to your tissues, you can you'll have better physical conditioning. So that's how he was cheating, and that's why it was pretty hard to detect, because it was his own blood. The other ways people have cheated, which is taking erythropoietin, which stimulates red blood cell production. And there's a real nice chart in your lecture text that shows the negative feedback cycle of erythropoietin. So definitely take a look at that. So there are some really big dangers. So it can lead to an embolism, it can lead to a stroke, or it can lead to heart failure, because you have that increased blood volume, which is really tough on the system. It's a lot of pressure on the system. So now let's talk about anemia and the causes. So there's three main categories you can put anemia into for causes. So inadequate erythropoiesis or hemoglobin synthesis. Erythropoiesis, remember that's red blood cell production. So what could be the causes? It could be caused by kidney failure and insufficient erythropoietin. So we just talked about erythropoietin just right before this. Iron deficiency, so nutritional deficiencies, vitamin B12. So this is another nutritional deficiency. You have hypoplastic anemia, slowing of the red blood cell production, or you have aplastic anemia, complete stop of the red blood cell production. You can also have anemia from bleeding. If you're bleeding out, if you lost a lot of blood, that will make you anemic. Also, hemolytic anemias from red blood cell destruction as well. Now let's talk about the white blood cells, the leukocytes. So leukocytes, so leuko means white, and then cytes means cell. These are actually, it's kind of misleading. So when you look here, well, it's not really misleading, but when we look at our slides, they're going to show up as these purple, these cells with these purple nuclei, and they don't normally look like that. That's just after we've stained it. So normally they're just clear. They're very clear, kind of white-like. That's why they're called white blood cells. So they're the least abundant formed elements. You look at the slide up here, and you can see tons of erythrocytes. Those are the most common formed elements. Then you have some platelets here. So these are the least abundant formed elements. These are going to protect against infections, microorganisms, and other pathogens. They're going to only spend a few hours in the bloodstream before they're going to migrate to connective tissue. One thing we need to talk about is granules. We have granulocytes and agranulocytes. We have two divisions of white blood cells, and so all white blood cells have lysosomes called nonspecific granules, but the granulocytes, those neutrophils, eosinophils, those are going to have specific granules that contain enzymes and other chemicals employed in defense against pathogens. So they're going to have those specific enzymes. We have two categories, granulocytes and agranulocytes. So the granulocytes, remember I said they are PHIL'd, they're filled with granulocytes, so neutrophil, eosinophil, and basophil. So these are your granulocytes, and that'll help you remember, keep that straight. And then you have your agranulocytes, your lymphocytes, and your monocytes. And remember, I have that mnemonic that helps you remember the different percentages. So that's in that other video. Make sure you take a look at that. But we're going to take a look at each of these individually. Another activity we do in lab is we do a differential, white blood cell differential count. Basically, we have our microscope, we zoom in to this point here, and we count how many white blood cells and what type of white blood cell that we see in this field of view. Then once we're done with that field of view, we move it down, count here, here, and then we go up and down until we have enough to make percentages. So if we saw, let's say we saw out of 100, we would see about 60 neutrophils, and that makes sense because that's kind of that's what you're supposed to see. And then out of 100 white blood cells that you counted, you'd see about 30 lymphocytes, and out of 100, you would see about 8 monocytes and about 3 eosinophils. So this is that mnemonic. Make sure you watch that video on the mnemonic. That'll help clear that up for you. Just, well, it'll make you really understand it, and you'll never forget it. Now we're going to focus on the granulocytes. So we have the neutrophils, the eosinophils, and the basophils. So I really want you to know, so neutrophils are aggressively antibacterial, and a rise in the number of neutrophils is called neutrophilia. And this is going to be in response to bacterial infection. So if, for example, if you have a question like with a differential white blood cell count, the neutrophils are highly elevated, or it gives you a number that they're elevated or something like that, then you would think it's a possible bacterial infection. So you need to be able to figure out if one of these leukocytes is elevated in a differential count, that what kind of an infection or what do you think is going on. So neutrophils, antibacterial, so bacterial infection, so it's going to be elevated in a bacterial infection. Eosinophils are increased in numbers in parasitic infections. Also some collagen diseases, allergies, and some other conditions as well. Basophils are very active in the allergic response. And here you can see the different white blood cells here. Here are the neutrophils, the most numerous ones. Then you have your basophils, eosinophils, sorry, and then you have your basophil right there. Now we're going to talk about the agranulocytes. So these are lacking the granules. So lymphocytes, these are going to be increased in numbers in diverse infections, many different immune responses. So there's a few different examples there. Monocytes are going to be increased in number in viral infections. So monocytes are going to be increased in viral infections. That's important because we want to figure out if it's going to be it's a bacterial infection or a viral infection. And you're going to see an increase in monocytes with that white blood cell differential count if it is a viral infection. So, and then remember we were talking about bacterial, and neutrophils are going to be elevated in a bacterial infection. So make sure you have those down. Here you can see these white blood cells. So there's a lymphocyte, there's a monocyte. So the monocyte's really big, and it's got that kidney-shaped nucleus right there. Now let's talk about white blood cell disorders. So leukopenia would be a low white blood cell count, below 5,000. I don't need you to remember the specific numbers. Just know that leukopenia would be is low white blood cell count. And leukocytosis would be a high white blood cell count. So let's go back to here. So some causes of leukopenia would be radiation, poisons, infectious diseases, effects, elevated risk of infection. So if you have, which makes sense, because if you don't have your white blood cells fighting infections, you're going to have increased risk of a serious infection. And leukocytosis, again, high white blood cell count above 10,000. Causes could be infection, allergy, or disease. So differential white blood cell count identifies what percentage of the total white blood cell count consists of each type of leukocyte. So that's why, remember, it's important to remember which ones are elevated during a bacterial infection or a viral infection, so you can identify, if you can identify which white blood cells elevated, you can narrow it down to what kind of an infection. So leukemia usually produces a very high number of circulating leukocytes. So you'd think that that'd be a good thing to have a lot of leukocytes in your system, these white blood cells, which are going to fight against infection a big factor in your immune system here. But it's actually not a good thing because of these white blood cells are actually immature white blood cells. So they're not mature ones, so they're not going to fight infections nearly as well or very much at all compared to these mature ones up here. So here's a normal blood slide here, a more normal one, and then here is a blood slide of leukemia, and you just have a ton of these white blood cells here, but they're very immature, so they're not prepared to fight infections, which is a problem. Especially these patients are going to be very prone to opportunistic infections or any infections. One last thing I wanted to talk about. So what we've been talking about hematocrit, we've been talking about hemoglobin concentration, we've been talking about uh, differential white blood cell count. But I just wanted to just summarize this. So the complete blood count includes all of these. It includes the hematocrit, it includes the hemoglobin, it includes the total red blood cell count, the total immature red blood cells, the total white blood cells, and the number of platelets as well. And it also includes that differential white blood cell count, which we went over, and also it goes over red blood cell size and the hemoglobin concentration per red blood cell. So that's what a CBC is, a complete blood count includes all these different values.